This video will explore Chapter 6, Energy, Enzymes, and Biological Reactions. Remember, Chapter 6 and Chapter 7 lecture notes will be covered on Exam 3. Use the study guide on Blackboard to prepare for Exam 3. By the end of this video, and subsequent review of Chapter 6, you should be able to discuss the learning objectives, using information from Chapter 6. The term thermodynamics will be defined later in the video. For now, this figure illustrates three different types of systems in thermodynamics. Living organisms are considered an open system. This means that living organisms have the ability to exchange both energy and matter with the surroundings. In biology, energy allows a living organism to carry out the processes of life, required to sustain life. There are two major types of energy: potential energy and kinetic energy. Potential energy represents stored energy, while kinetic energy represents the energy of motion. Thermodynamics is the study of the energy conversions that take place in biological units. The two laws of thermodynamics provide a way of describing energy dynamics in living systems. Energy, which can exist in many forms, moves throughout components of the biosphere. Energy is utilized and transferred to promote life on Earth. Unlike plants and other photosynthetic organisms, animals cannot generate energy directly from sunlight. Animals must consume plants, or other animal organisms, for energy. The energy stored in glucose can also be released through cellular respiration. This process allows plant and animal organisms to access the energy stored in carbohydrates, lipids, and other macromolecules through the production of ATP. This energy is needed to perform cell functions, such as DNA replication, mitosis, meiosis, cell movement, endocytosis, exocytosis, and apoptosis. In photosynthesis, the energy is supplied by the sun. Light energy is absorbed by cells in plant leaves and converted to chemical energy. The chemical energy is stored in the form of glucose, which is used to form complex carbohydrates, necessary to build plant mass. The first law of thermodynamics, also known as the law of conservation of energy, states that energy can neither be created nor destroyed. It may change from one form to another, but the energy in a closed system remains constant. The second law of thermodynamics states that when energy is transferred, there will be less energy available at the end of the transfer process than at the beginning. Entropy is the measure of disorder in a closed system. Entropy increases as energy is transferred. Take the time to review this sample Exam 3 question. If you have any questions about this slide, contact Dr. Flowers via email. Two major types of reactions take place in living organisms: exergonic reactions and endergonic reactions. Exergonic reactions are energy-releasing chemical reactions. Endergonic reactions are energy-requiring chemical reactions. Catabolism is a fancy word for breakdown, or destruction of biological molecules. Catabolic reactions are exergonic, or energy-releasing reactions. Catabolic reactions result in the breakdown of polymers to monomers. The image on the bottom of the screen illustrates a catabolic reaction. Here, you can clearly see that a large molecule is being broken down into a smaller molecule. This slide also shows you that there are various synonyms for the word catabolism. Catabolism, degradation, hydrolytic, and exergonic all mean the same thing. The opposite of catabolism is anabolism. Anabolic reactions are synthetic reactions that involve the binding of large biomolecules from smaller molecular subunits. This slide shows you that there are various synonyms for the word anabolism. Anabolism, biosynthetic, dehydration synthesis, and endergonic reactions all mean the same thing. This figure shows a graphical representation of an exergonic reaction and an endergonic reaction. ATP hydrolysis, or ATP breakdown, will be discussed in later slides. For now, understand that the conversion of ATP to ADP, with the subsequent release in usable energy, is necessary to power energy-requiring reactions. The chemical structure of adenosine triphosphate is shown here. The top part of this figure shows the ATP molecule. ATP is made up of an adenine, a nitrogen base found in RNA, and DNA, a ribose, a 5-carbon sugar, and 3 phosphate groups. The bottom part of the figure shows how cells extract energy from ATP. Specifically, the bottom part shows ATP hydrolysis. It shows that the conversion of ATP to ADP involves the cleavage of the outside, or third phosphate group. When ATP becomes ADP, energy is released. The energy that is released is used to power cellular activities. ATP is used to power cellular activities. When a cell uses chemical energy to perform work, it couples an exergonic reactions with an endergonic one. The ATP-ADP cycle shown on this slide demonstrates how exergonic reactions produces energy that in later reactions are used to drive endergonic reactions. This figure shows the unique relationship between exergonic reactions and endergonic reactions. It also shows that ATP breakdown and ATP synthesis are necessary aspects of metabolic reactions. This figure shows the importance of ATP hydrolysis, or ATP breakdown, to help drive biological reactions. This figure shows that when coupled with ATP hydrolysis, the glutamine synthesis reaction is possible. The next two slides show how cells use ATP energy to power cellular work. Before we discuss the enzyme-catalyzed biological reactions that require breakdown, or synthesis of ATP, we must first discuss the topic of activation energy. Quite simply, this is a type of energy that must be overcome, or reduced, prior to the activation of a biological reaction. During this presentation, we will define and illustrate the activation energy barrier, using pictures and graphs. Activation energy is defined as the minimum amount of energy required by a reactant in order to convert into a product. Alternatively, activation energy is the minimum amount of energy needed to activate molecules so they can undergo a chemical reaction. The top half of the figure provides a biological example illustrating the activation energy barrier in the oxidation of glucose. It shows that in order for the conversion of reactants, glucose, and oxygen, to products, that a minimal amount of activation energy is necessary. The bottom half of this figure provides another illustration of activation energy. Here, the activation energy is represented as a bump, or hill. Once this bump is reduced, or lowered, it will be much easier to roll the rock down the hill. In terms of biochemical reactions, once the activation energy is reduced, a biological reaction can also proceed. It is important to know for exam 3, that enzymes, a topic we will discuss later, are used in living organisms to reduce the activation energy and allow reactions to occur much faster. This figure, and the next two slides, clearly show you the difference in activation energy size, in the presence, or absence of an enzyme. Note, that when an enzyme is present, the activation energy is lower. This slide provides a basic definition of an enzyme. It also provides a cartoon diagram showing the substrate and a critical location on the enzyme, the active site. Enzymes, which are typically proteins, are essential molecules that are required to accelerate that rate at which key biological reactions occur. Again, enzymes work by decreasing the energy barrier, or activation energy, that slow down reaction completion. Once the activation energy is reduced, reactions occur much faster. As this image demonstrates, the substrate must bind to the enzyme, at the enzyme's active site. For exam 3, it is important that you understand the definition of the terms enzyme, active site, and substrate. The physiological significance of enzymes to humans is enormous. There are many human diseases, and human deaths each year, associated with enzyme deficiencies, or enzymes, that just don't work like they are supposed to. Enzymes are biological catalysts that speed up the rate of a reaction, without being changed in the process. Enzymes bind substrate, at a particular site on the enzyme, called the active site. Following production of the enzyme substrate complex, the substrate is converted into a specific product. There is an affinity, or specificity, for enzyme, and a specific substrate. This is an excellent image of an enzyme, a substrate, and an enzyme's active site. The active site is the region of an enzyme that interacts with the substrate. Remember, a substrate is any compound with which an enzyme reacts. There are two prevailing models that help scientists Understand the nature of the interaction between the substrate and the enzyme. They are the lock and key model, and the induced fit model. The lock and key model was the first proposed model, and suggests that the substrate and enzyme have a predetermined conformation that does not change, similar to a lock and key. In this model, the lock refers to the enzyme's active site, and the key refers to the substrate. This model is good because it is simple and easy to understand. However, it was later determined that while some enzymes and substrates fit this model, most enzymes adhere to the induced fit model. The induced fit model suggests that the active site is not rigid, but flexible, and that it basically conforms to the shape of the substrate. In a sense, the substrate induces, or makes the active site match its shape, to form a very strong association. This is an excellent image of an enzyme, a substrate, and an enzyme's active site. The active site is the region of an enzyme that interacts with the substrate. Remember, a substrate is any compound with which an enzyme reacts. Take the time to review this sample exam 3 question. If you have any questions about this slide, contact Dr. Flowers via email. This slide shows the 5 major steps involved in an enzyme's ability to convert a reactant or substrate into a product. As you will easily see, in the next few slides, the formation of the enzyme-substrate complex is an essential event in the conversion of reactants to products. As I said before, enzymes speed up the rate of a reaction. The current slide shows you how this is accomplished. Essentially, enzymes act as matchmakers, bringing substrates together to facilitate a reaction. In step 1, substrate binds to the enzyme, forming an enzyme-substrate complex. Following, the formation of the enzyme-substrate complex. The products are released from the enzyme's active site, allowing the enzyme the opportunity to initiate another reaction. Review the 3-step catalytic cycle of enzymes for exam 3. This last image provides the illustration that matches the 5-step process described earlier. An enzyme is defined as a biological catalyst that speeds up the rate of a reaction without itself being used or changed in the process. This image validates that definition. Notice how the enzyme in the illustration represented by the blue shape looks the same in step 1 and step 5 of the process. This image shows that an increase in enzyme concentration leads to an increase in enzyme activity, as long as the substrate concentrate is at a high enough level. All biological molecules have a preferred temperature, pH, and microenvironment, at which they require to function normally. Any change in the temperature range, or pH range, or change in another parameter, will cause the molecule to function poorly. Just like other biomolecules, the temperature, pH, and other functions can negatively affect the enzyme's performance in a physiological setting. When an enzyme encounters a high temperature, or abnormal pH, then the enzyme may denature. The images shown on this slide show what normal proteins look like, and what denatured proteins look like. Denatured enzymes are inactive and non-functional. This slide lists the 4 major factors that influence the activity of an enzyme. The 4 factors that alter the functionality of an enzyme are temperature, pH, substrate concentration, and the presence or absence of an inhibitor. Most human enzymes function properly at 37 degrees Celsius. At high temperatures, enzymes, which are mostly proteins, unfold or unravel. The unfolding or unraveling of a protein is called denaturation. A picture of a denatured protein is shown on the next slide. An enzyme typically has an optimal pH, at which it is most active. At pH values above or below the optimum, the rate of enzyme activity drops off. At extreme pH values, the rate drops to 0. Most human enzymes function properly at pH 7. At high or low pH levels, enzymes denature. Enzymes typically work fine when there is an abundance of substrate present in the environment. However, when there is an excess of substrate in the environment, saturation is achieved. Saturation is the state at which no more substrate can be added to the active site of an enzyme. This slide shows the temperature at which enzymes are active and inactive. Using this graph, we can clearly see that the optimal enzyme activity is 37 degrees Celsius. Visible effects of environmental temperature on enzyme activity is shown in the Siamese cat. The fur on the extremities, ears, nose, paws, and tail contains more dark brown pigment, such as melanin, than the rest of the body. This is because an enzyme controlling melanin production is much less active in warmer body regions, so dark pigment is not reduced, and fur color is lighter. Take the time to review this sample exam 3 question. If you have any questions about this slide, contact Dr. Flowers via email. Additionally, the pH of the microenvironment impacts enzyme activity. All enzymes have an acceptable pH range. Within this range, enzyme activity is normal. If an environment is too acidic or too basic, the enzyme, which is a protein, can denature. Stated differently, when hydrogen ion concentration in the medium is changed drastically, the protein's three-dimensional shape is altered. Acids and bases alter a protein's three-dimensional structure, because hydrogen ions and hydroxide ions compete with hydrogen bonds and ionic bonds in the enzyme, resulting in the enzyme's denaturation. This figure shows you how pH and temperature affect an enzyme's ability to function normally. As shown in this figure, initially adding substrate to an enzyme increases enzyme activity. However, At some point, the enzyme reaches a saturation level. Saturation is a condition in which the active site on an enzyme is occupied by the substrate, or product at all times. Saturation is illustrated by a flat line on an enzyme curve. Again, at the saturation level, the active sites on all of the enzyme molecules are filled. At this point, the enzyme's activity decreases. There are two major types of chemical inhibition that affect enzyme activity. An inhibitor is something that inhibits, or stops something from working properly. Competitive inhibitors, and non-competitive inhibitors, are designed to reduce the correct, or normal substrate from binding to the active site. During competitive inhibition, a chemical substance binds to the active site, to prevent the precise binding of a unique substrate to an enzyme's active site. During non-competitive inhibition, a chemical substance binds to another site, not the active site. Binding to another site on the enzyme causes a conformational change to the active site, thereby preventing the precise binding of a unique substrate to an enzyme's active site. Both competitive and non-competitive inhibition prevent enzymes from working. The next two slides show excellent illustrations of competitive inhibition. From these two images, you can clearly see that the competitive inhibitor is binding to the active site, blocking the substrate from binding. The next two slides show excellent illustrations of non-competitive inhibition. From these two images, you can clearly see that the non-competitive inhibitor is binding to an allosteric site, not the active site. Binding to the allosteric site distorts the active site, preventing the substrate from binding. This figure better illustrates how the binding of a non-competitive inhibitor alters the conformation of the active site. Alteration of the shape of the active site prevents the substrate from binding to the active site. Non-competitive, or allosteric inhibition, plays a role in a biochemical reaction control mechanism called feedback inhibition. Feedback inhibition is also called end product inhibition. Feedback inhibition is the inhibition of an enzyme in a particular pathway by the accumulation of the end product. Stated differently, the end product is used to allosterically inhibit enzyme activity. The image shown on the right represents the many thousands of biochemical or metabolic reactions that take place in your cells every second of every day. Prior to exam 3, be able to explain this image on this slide. Collectively, these biological reactions are responsible for energy production, energy utilization, and biomolecular synthesis and biomolecular breakdown. This image shows an overview of metabolism. It shows the relationship between anabolic reactions and catabolic reactions. Take the time to review this sample exam 3 question. If you have any questions about this slide, contact Dr. Flowers via email.